Thank you, Dr. Stover. It's my pleasure to introduce Dr. Saravanomuthu, who I know for the last 10 years. And he's the founder of ONS Bio, a strategic distribution partner for ROKIT Healthcare, INVIVO bioprinters, and Humethane bioinks. Dr. Saravanomuthu obtained his PhD in biology from the University of Hanover in Germany in 2004, and he did an extensive postdoctoral research training at the National Institute of Health in Bethesda in cell molecular and developmental biology. Currently, he's passionately pursuing popularizing the 3D bioprinting technologies and its adoption by life science researchers. Since 2012, he also serves as the Director of Business Development and Sales at BioBasic Life Science Research Products Company, and a leader in serving science. Today, he will speak on the topic of bioprinting in tumor biology, recent trends in 3D tumor microenvironment modeling. Over to Dr. Saravanomuthu.
Thank you, Dr. Shankar, for the introduction. And thank you, Dr. Stover and Dr. Ramaswamy, who is not here, but will soon join, for the opportunity today. I'll keep it brief. And I understand that most of you in the audience are relatively new or just planning to look into 3D bioprinting as part of your workflow. So it's good because I have more introductory type of a lecture planned today. And the idea is to get you introduced into some of the basics and then kindle interest based on the publications, so that you can debate and discuss during this Q&A or later on offline about ways and means in which you can adopt this powerful technology into your cancer research workflow. So thanks again for making it here today. I will turn off the video just that it's less distracting for myself as well as the audience, and we can all focus on the presentation. All right, as Ishwar mentioned, I am the founder and CEO for ONS Bio, which is the authorized distribution partner in the US Midwest for ROKIT Healthcare. And today I'm joined by our partner from the West Coast, Indicate Technologies, represented by my good friend Chris Kamimura here. And together, ROKIT, ONS Bio, and Indicate, we have this vision and mission to popularize 3D bioprinting and bring awareness among the scientific research community, the life science research community, about the power and potential of this technology for various disciplines of research. And hopefully this talk will kind of impress upon the same idea to you all and its relevance to use in 3D tumor modeling, which might be of interest to you all, I presume. So ROKIT Healthcare is a personalized regenerative medicine company headquartered in Seoul, South Korea, and with customer base and presence all over the world. And the company focuses on optimizing the power of 3D bioprinting in life sciences research and in the development of novel therapies using the same approach. My goal today, as I mentioned before, is to introduce 3D bioprinting and its applications and drive home the relevance to studying tumor biology and 3D tumor microenvironment modeling. My talk will be in kind of three parts. First, I'll give you a basic overview of what bioprinting is. Second, I will walk you through some case studies demonstrating how users of ROKIT's INVIVO bioprinting platform are using bioprinting to create really complex and reproducible models of 3D tumors for studying tumor physiology, biology, and in drug screening assays as well. Third, I will give you some details of the bioprinting packages we can offer for you to start using this powerful technology in your own research workflows right away. So let me first set the scene at a higher level to explain the driving force behind the increasing use of bioprinting technology in cancer research. We all know that three-dimensional cell culturing technologies lie somewhere between the traditional 2D cell culture and the animal models. Now, many of us can easily agree that 2D cell cultures are popular, easy to use, but they are not adequately representative of the in vivo human physiology or human tissue architecture. On the other hand, the animal models may be more biologically relevant, but they are expensive and require long ethics approval. In some cases, you simply can't find animal models, suitable animal models. So for this reason, 3D cell culture model offers an attractive alternative than anything that can better represent human tissues, while still being able to be executed at a relatively low cost with high repeatability. Now, a very traditional model of 3D cell culture technology currently widely used in cancer research, it relies on the monoculturing as well as co-culturing of cancer and stromal cells in a predefined three-dimensional hydrogel matrix. And the matrix is often matrigel or collagen or PEG hydrogels. Now, in this slide, what you see here is a representation by pictures on the left-hand side that of the 3D regular cell culture. Now, there has been quite some limitation, I should say, in the choice of the matrix biomaterial that can be used. And it has not been easy to architecturally position cells within the matrix in a way and manner that you need, because most of these systems are kind of a self-assembly systems. So you don't have much of a control over how and where the cells get placed. Now, against these challenges, 3D bioprinting is rising to represent a kind of a technology shift in how we do these 3D cell culture modeling. The 3D bioprinting is defined as a computer-aided biomanufacturing technology that allows for free-form printing of hydrogels and cells to fabricate 3D tumor tissues that have very high architectural complexity, but they can also make use of various biomaterials all at once. So as you can see here, going back to the same picture from the previous slides, on the right-hand side, what you see here is the glioblastoma 3D vascularized patch development. And here we take glioblastoma as an example. So the bioprinting technology allows the researcher to create an ideal mix of biomaterials, such as alginate, fibrinogen, gelatin. There are a whole bunch of those out there. And then to be able to print a patch of vascularized tissue and see tumors on top of it to create a tumor microenvironment. Now, this is in contrast to the left side here, which shows dispensing of co-cultured aggregates in sphere-like formations in a predefined matrix. So using bioprinting now, you can more precisely define the dimension, composition, and shape of a tumor microenvironment. And one can place different types of cells and different types of hydrogels in positions you want. Thereby, it enables better interactions between the cells and overall gives a better mimic, or it can better mimic the biophysical properties and even mechanical stiffness of a three-dimensional tumor tissue. So in short, 3D bioprinting offers a way to combine many different components to work together in one single platform to achieve the most ideal three-dimensional fabrication of human tissues, closest, I would say, to their actual anatomy. Now, if a bioprinting platform like this can be translated off the bench into the hospital, then one can imagine that it could offer a complete paradigm shift in the workflow for cancer tissue studies. And here what you see is a comparison of the timeline that one needs between a conventional method, such as the PDX model or organoids harvested from tumor biopsy, versus what a bioprinted approach can provide, a difference of 5 months versus approximately 0.5 months. And so using patient's own cells and patient-derived hydrogel matrices, the ability to print patient tissues on demand can reduce the overall time and cost of tumor modeling processes, and it can increase the biological relevance of drug screening assays with less dependence on ineffective or expensive models. Finally, with the aid of the automation, bioprinting offers to kind of broaden the project scope and workforce diversity within cancer research centers by potentially reducing an otherwise manual method of experimentation. All right, so now that we understand the backdrop of why bioprinting is rising to attention in cancer research, let's talk about the various bioprinting strategies that are being used in 3D tumor modeling these days. So current bioprinting techniques can be broadly categorized into 5 types. And those are inkjet bioprinting, laser-assisted bioprinting, extrusion-based bioprinting, the Kenzan method, and the stereolithography-based bioprinting. Inkjet and laser, as you can see, they use electrostatic pulses or laser as the energy source to deposit biomaterials onto a substrate. Whereas the extrusion-based bioprinting uses mechanical or air pressure to force the material, hydrogel combined with cells, say bioink, out of a nozzle, much like how you would extrude the toothpaste out of a toothpaste. So the Kenzan method relies on skewering already developed cell aggregates onto tiny needles in 3 dimensions, while the stereolithography-based bioprinting, it uses UV or visible light to polymerize the resins into solid 3D structures. Now, each of these technology has its own pros and cons in terms of resolution, material diversity, the speed, the sensitivity to cells, and how the cells are behaving, whether they are happy during the print process. Now, all in all, one can say that in today's market, extrusion-based bioprinting is the most common and widely used, widely researched technique for bioprinting. So today, we will focus on this technique in the rest of our discussion. So the delicate nature of substances like cells, hydrogels, and the elastic polymers used in 3D bioprinting, it means this can result in fragile, mechanically unstable constructs after you print. So therefore, in order to overcome this and successfully print tissue constructs with biologically relevant durability, we need to be aware of several methods of stabilization that are often applied during and or after the printing procedure of not just 3D cancer tissues, but any tissue for that matter. So the first of these stabilization methods is the bioink cross-linking techniques, which is a direct cross-linking of soft hydrogels or hydrogel-based polymers rather, using either or several of these different chemistries, say ionic, enzymatic, radiation, or thermal means. So examples of each of these chemistries is kind of given here. You can see, for example, the alginate is often stabilized using calcium cations. So that's an ionic method. And on the other hand, if you take the enzymatic approach, the good example would be fibrinogen thrombin that can form a fibrin glue, and so on and so forth. In case of radiation, often we refer to the use of UV or visible light to cross-link a polymer. And thermal methods rely on the use of changes in temperatures from heat to cold to stabilize. And one example would be the case of collagen cross-linking between 37 degrees and 4 degrees temperature changes. The second one is the extrastructural support method. This is the most elementary supporting strategy based on the fabrication of a framework around the construct, which stabilizes and holds the extruded bioink and provides a platform for the printed structure to lean on so that it doesn't collapse on itself. So here what you see in part A is an example of the engineered cartilage tissue printed with decellularized cartilage extracellular matrix bioink in combination with a PCL, polycaprolactone framework. And in B here, what you see is an example of an engineered osteogenic tissue printed with the same PCL, but kind of a soft hydrogel type pluronic PF127. The third one is called the intrastructural support strategy, in which one ink is used to create a soft supporting structure called a matrix, while the other ink can be used as a fugitive ink. By fugitive ink, what we mean is that the ink is used to create a hollow channel, like in this case, you see, that serves as kind of a sacrificial material, which can be dissolved post-printing, and then that would leave behind channels which can be used to create vascularized channels, which can be eventually filled with, say, the HUVEX or endothelial cells that can infiltrate, perfuse, and form the potential vascular tissue within the printed tissue. Finally, we have the fourth and last fabrication strategy, which is called the precasted support. Basically, a support bath is used, like a slurry, which provides a semi-solid medium onto which your water-rich, low-viscosity bioink can be printed, overcoming the limitation of the of printing the same low-viscosity ink on a flat air interface. And here is an example of the same, where complex biological structures can be printed using bioinks such as alginate, fibrin, or collagen, type 1, or matrigel, by printing the hydrogel in a support bath of gelatin microparticles, as you see here. So how are these basic strategies of extrusion bioprinting utilized in real life applications in tumor biology? That's the question for today, right? So to walk you through this, I have compiled a couple of case studies. There are many out there, but in the interest of time, we'll just touch upon a few so that you get the general idea of how people have been using and how you could also think about ways to use for your own research. And if time permits, we can brainstorm some ideas now or later to see how you can adopt these things into your workflow. So all of the studies that are indicated here with Dr. INVIVO on the top, that means they are all publications written by the users of Dr. INVIVO bioprinting platform from ROKIT Healthcare. Of course, I do have examples of use of bioprinting in tumor biology from other platforms as well, but we'll get to that when we get to those slides. So in this study, the authors created a three-dimensional cell scaffold. I'm not going to go into the great detail of these studies. That's something that you can always catch up by reading the publication, but I'm just going to give you the overview of the study and the advantages and the benefits that bioprinting is bringing into this study. So in this study, the authors created a three-dimensional cell scaffold using gelatin methacrylate, which is called GELMA, and compared the cell survival and protein expression in 3-dimension versus traditional 2-dimensional cell cultures. Now, they were first able to show that in cell viability assays, cells exhibited very high viability in GELMA bioink, because that answers the first question, are cells happy during the process? Yes, they are. And this was examined over multiple days after printing. So viability is not a problem. Then they were able to show that not only did these 3D cell cultures show higher cancer cell proliferation rates than the 2D cell cultures, they also showed higher cell-to-cell interactions through the secretion of E-cadherin and N-cadherin. Assessment of the effects of drugs for bladder cancer, such as rapamycin and BCG, showed that the effect in the 2D cell culture environment was more exaggerated than that was observed in the 3D cell culture environment. So, in short, the authors conclude that the bioprinting technique can be used to create cancer cell-like environment for a drug screening platform with greater biological relevance than the 2D cell culture system. Now, taking a step further from the previous publication, this second study shows how to add compositional and dynamic complexity to a 3D bioprinted cancer tissue by creating extracellular matrix microenvironment and vasculature. So in this study, the authors have created a complex multicellular scaffold consisting of endothelial fibroblast mixed with the glue of alginate, gelatin, and fibrinogen hydrogels. Now, this printed vascularized tissue was then seeded with U87 derived glioblastoma spheroids, and the U87 spheroids cultured in the bioprinted matrix and enriched in vascular structure. It showed increased expression of the epithelial mesenchymal transition markers such as N-cadherin and vimentin. And it indicates a kind of a cross influence among the between the tumor and the normal cells. So this model was treated with the anti-cancer drugs temozolomide and the anti-angiogenic molecule sunitinib, and it kind of showed the synergism of their combination to affect both the tumor spheroid size as well as the vascular network. So in conclusion, what I'm trying to say here is that the bioprinting can be used to produce large open vascularized tissue very easily and rapidly compared to any other biofabrication method. And it can create an in vitro vascularized tumor microenvironment with controllable tumor spheroid sizes. So now we have introduced, we've come from the 2D to 3D cell culture, and then we introduced some vasculature and dynamic complexity to the model. Now, as with one step further, now in this case study 3, what you're going to see is that there are more complicated things that can be done using the bioprinting. So just to give you another example from external sources, what we have here is the increase in the complexity level of the model. So in this third study, what we're seeing is how a high throughput bioprinting and patient-derived tumor organoid can be combined to create a drug screening platform. So here, what has been done is the tumor organoids were combined with patient-derived cancer-associated fibroblasts and the endothelial cells to create what is called assembloids. And these assembloids were further printed with Dr. INVIVO bioprinting platform in a very consistently controlled, approximately 10 microliter sized droplets in 96 well formats in order to develop a patient-specific, tumor-specific drug screening assay. So the authors were able to demonstrate that given the high throughput system and the ability to print tumor assembloids in precise volume, the bioprinter has the potential, or bioprinting technique has the potential to revolutionize in-hospital precision medicine-based testing platforms for cancer tissues. And this is just one example. So just to give you a few more examples from external sources. By external sources, I mean these were not done using the ROKIT's INVIVO platform, but these were done with Dr. INVIVO, with other systems. What we have here is, but one thing in common though is that these were also done using the extrusion type printing, the micro extrusion type printing, which is the same technique that the ROKIT platforms also use. So in this study, we see advantages of bioprinting in reconstitution of a tumor microenvironment by creating a gradient of layers representing different nutrients and gas levels and or biomechanical stiffness. So here we see the glioblastoma on a chip that was established by the printing of patient-derived glioblastoma cells co-cultured with endothelial cells on a decellularized extracellular matrix environment. In this model, a cancer concentric ring structure, as you can see here, it maintains a radial oxygen gradient to mimic the in vivo tumor-like microenvironment. Now, this model is known to have shown clinical significance by reproducing the patient-specific resistance to concurrent chemoradiation and the drugs. Now, taking cues from that publication, we tried to replicate the printing in-house using the INVIVO platform, and we were able to replicate that in this little demonstration that you see here, where we were able to test the hydrogels to show indeed that such a study can be performed with our own platform, where the gradient can be created not only horizontally, but also vertically. Another external case study here, which is probably the last one I'll be talking about, is the example where the use of bioprinting PCL scaffolds and GELMA-based scaffolds was used to co-culture breast cancer cells and bone cells in order to mimic the activity of breast cancer cells metastasizing onto the bone tissue. Now, this is another example of tumor microenvironment modeling using 3D bioprinting that makes use of a makes a hybrid use of hard polymers like PCL, hydroxyapatite, and PLA to create bone-like tissue as the substrate, and using the soft polymers like gelatin, collagen, Metrogel to create the soft cancer tissue. So you've got two different types here, the hard as well as the soft. Once again, we did try replicating those using our platform, and we were able to do that. So far, we have discussed the basics behind the technology of bioprinting, the reasons that it is rising to attention in cancer research, and the unique values and opportunities it offers to research aimed at advancing our knowledge and understanding of tumor microenvironments. So at this juncture, I think I should emphasize to you, the audience, that the most powerful feature or fact about bioprinting or 3D bioprinting is that it is a platform technology, and that its application in cancer research is just one of many possible applications. So in fact, if you look, the applications range from all kinds of disciplines, from tissue engineering to drug discovery to non-animal testing, chemical safety testing, cancer microenvironments, organ and tissue regeneration for transplantation, so on and so forth. So this is indeed a very powerful, robust, widely applicable technology, not necessarily restricted to any one area of research. So just to impress upon that fact that it is widely applicable, I will quickly run through the next few slides to give an idea of some of the other applications. This is just a very, very small snapshot of the possibilities, but the reality is there are so many, and we can go on and on. But in the interest of time, I'll just restrict it to these few examples just to give you an idea. This is, of course, the kind of the holy grail for bioprinting, because at the end of the day, we want to be able to print organs that can be regenerated from patients' own cells and used to create functional organs that can be actually put back into the patient. So that is the holy grail. Now, there are applications in terms of research, applications in nerve, understanding nerve regeneration. This is one example of where augmented peripheral nerve regeneration was studied through elastic nerve guidance conduits that were 3D bioprinted. And here is an example of kind of an innovative drug delivery and patient info delivery device that was 3D bioprinted to it has a QR code on the patch, drug delivery patch, and that can help in enhancing the patient compliance to prescribed drugs, and also it can enhance drug solubility for patients with difficulty swallowing. So this is just another example of the varied applications possible. And this is one other example of application in developing a biosensor, not just using mammalian cells that we all are generally used to thinking as the application for bioprinting. Here, in this case, it's a microbial study. So it's not restricted to only cells. So that's the point I'm trying to drive home here. And so on. There are many other applications in bone, in any tissue of, that you can think about. So then comes the question, all right, so we've seen a lot of different potential examples and all that. So what is in it for you? So ROKIT Healthcare offers a global infrastructure of 250 plus bioprinting users across 50 plus countries, spanning academia, industry, governments, and they represent various stages and disciplines of biomedical research and development market. And they have produced over 100 plus publications so far, offering evidence-based usage of bioprinting. And these are some of the key users, if I may say so. And these are some institutions across the world that have the ROKIT INVIVO bioprinting platform. And in this context, I must mention that ROKIT Healthcare is also offering a bioprinting platform technology that comprises of not just the hardware, it also includes the software, the biomaterials, and the bio inks, which are a very important component of this whole workflow, and also the educational and training aspect that goes with it, with not just bioprinting, with any novel technology that's kind of cutting edge and still needs a lot of catching up to do. So first, let's take a quick look at the hardware options here. What you see here is the Dr. INVIVO 4D2. Basically, ROKIT offers two types of bioprinting systems, the Dr. INVIVO 4D2 and the Dr. INVIVO 4D6. And what you see here is the Dr. INVIVO 4D2, which was the world's first desktop bioprinter to have been commercialized for clinical regenerative treatment. And it is also the world's first clean chamber bioprinter equipped with a HEPA filter and UV for bioprinting with minimal risk of contamination. It is currently used by over 250 plus reference labs worldwide, with the 30 plus publications and international recognitions already. So it offers a hybrid capability, meaning it has multiple types of print heads, like the hot melt dispenser that is useful for melting pellets and powders and paste of very hot melting materials like PCL, so on, so forth. And then it has a filament extruder that is suitable for extruding the synthetic polymers like PLA or PCL, things like that. And then we've got the syringe dispenser, which is very important when it comes to being able to print bio inks and soft hydrogels with cell mixture. And this is again the same point that you can do multiple types of materials using the INVIVO 4D2 and across a wide range of temperature. So this wide range of temperature and pressure that the instrument can handle kind of makes it future-proof, because when you are embarking on a project like this, you have no clue what is going to come in future, what type of materials you may possibly want to work with. So this wide range of temperature and pressure that the bioprinter can handle, combined with the different print heads, gives you the versatility and the flexibility to experiment many different materials and then come up with innovations that are simply limited by only your imagination and not by the equipment. The Dr. INVIVO 4D6 is a relatively new equipment. This is the first of its kind, only FDA registered, and not to be confused with FDA approved. So one of its kind, FDA registered bioprinter that offers following unique characteristics, which help optimize tissue engineering and regenerative medicine research. It has an inbuilt cell incubator, because that's why we like to call it 4D printing, because printing is just one aspect, but then you need to have this temporal component and provide the suitable carbon dioxide and humidity for the cells, both during the process and post printing, to make them mature into something functional. And so we realized early on that there is a need to incorporate the cell incubation function right into the bioprinter. So that's out of that idea was born this INVIVO 4D6. Plus, it has a lot of other very medically relevant features, such as we all know that the human body is a curved surface. So any bioprinter that needs to be printing, say even in situ or trying to recreate human tissues, needs to have the ability to do curve printing. And so in that context, this has the ability to do in situ 4D printing in a curved fashion, which is not possible by many other printers out there. And it has 6 print heads for complex tissue fabrication with hydrogels, filaments, and polymers, pellets, and various FDA-approved materials as well. And it has complete particle control with circular airflow generation technology, which is more like a biosafety cabinet. And it has a very high throughput to a maximum of 384 well format for any tissue screening and assay development. All right, and now that we've taken a look at the hardware, I also kind of briefly touch upon the second of the ROKIT Healthcare offerings in terms of the whole extracellular matrix bioinks for versatile tissue engineering. You get a panel of versatile bioinks, because these are very important. No matter how great a printer you have, unless you have the right choice of bioink, extracellular matrix material, you cannot recreate the tissue the way you want to, in the way it should be to be physiologically relevant. So because of that, ROKIT has taken the effort to have a dedicated bioink development team. And so they offer versatile bioinks that can be useful with customization to different human tissue environments. And Humatine is a cell culture biomaterial supplement consisting of full native human extracellular matrix, useful for 3D bioprinting and cell culturing as well, if you so desire to get started. And it can also be useful for clinically translational studies, say wound healing or anti-aging type of studies as well. It's just a continuation of that. There are other materials like the INVIVO gel that are GELMA based and so on. So there is a wide range of hardware as well as the biomaterials that's available in the form of even kits that are ready to go in a tissue for a tissue specific research. And of course, as I mentioned, the last component, but a very important one though, is to have a robust educational and training outreach effort. In fact, today's effort is part of one such, the educational effort from ROKIT and its distribution partners to get people to realize the potential of bioprinting technology and how it is kind of shaping up to be the fourth big revolution in biomedical research and healthcare. So this is done through a series of events, workshops, and of course, very dedicated technical support for the existing users and future users of the systems, to help them experiment and to guide them in the experiments from the design phase all the way through the execution. So lastly, I want to conclude by saying that bioprinting represents a convergence of technology, convergence of knowledge, technology, and skills across various disciplines, from medical, engineering, pharmaceutical, data science fields. And this interdisciplinary and collaborative nature of the technology is kind of at the core of bioprinting's mission to develop new insights into cancer and other diseases, and as well as serve as and provide new tools and technologies and collaborative kind of a team-focused efforts to better treat, diagnose, and prevent disease. So if you have any questions about the application of bioprinting to your, or the potential for bioprinting to be applicable to your research, please do let us know. We'll be very happy to have that discussion with you. And I hope what we have talked today will give you some impetus to go explore about the possibilities to adopt in your own work. And I think at this rate of progress with bioprinting on a daily and monthly basis, it is possible to conceive that soon most of the journals will start asking for experimental data from bioprinted tissues as kind of a bridge between the preclinical research using 2D cell culture or animal models and the clinical research data. So that's all I've got to say today. Thank you very much for your time and attention. And I will be happy to take some questions now.
Thanks so much. I know that Eshwar, and certainly defer to you, Eshwar, in terms of thinking about applications. But I think sort of one of my questions, given potentially diverse interests of a couple of us interested in breast cancer, potential settings where multiple investigators or teams might be able to benefit from a sort of a single approach, or do you find that everyone sort of refines, to their specific interests?
No, I think as I mentioned, the power and potential of this technology is that it's more like a platform technology. So each of the groups that have interest, they can try their own approaches without having to have different platform or technology. So the single block of technology can provide and cater to not only the various approaches of breast cancer group here, but any other group doing anything, maybe cancer related or completely different, no problem. The way to think about this is more like your regular printer. I mean, that might be kind of a, pardon me if it's a lame analogy, but I like to think about it this way. You can have a printer, and with that printer, you can print an invoice, you can print a journal article, you can print a nice novel, or you can print a nice, a nice piece of letter. Now, each of these are very different in their purpose and intent, but all of this comes from the same. But this is a very simplistic analogy because it uses just one type of ink. But here you have variations at every possible component: the ink, the print head, then the architecture. So basically, it's just how much you understand your biological system. That is the key. If you knew very well about your tumor microenvironment based on the existing data from your own work and from the literature, then you can design in a computer-aided fashion a very complex architecture that is relevant for your purpose. And that may be different for somebody else, no problem. And then you're not restricted to using only one cell type or two cell types. You want to throw in 5 different cell types, no problem. So the short answer is each one can have, one single equipment can do many different things for many different people.
Gotcha. Thank you.
I hope that's what you asked. And that's
no, that was that. I think that exactly that you really got at the customizability of it, you know, and,
Yes.
and how modular it is, that it doesn't really restrict. So thanks.
It doesn't restrict at all. And that is the beauty here. More like our qPCR. I mean, one person could be using it to study gene expression, but then someone else could be using it for diagnostics. But at the heart of it, it's more like a platform technology.
Other questions? Eshwar, Eduardo, Ramesh, anyone else?
Yeah, I have a question.
Yes, please.
First of all, sorry, my camera is not working, but yeah,
No problem,
very interesting.
Eduardo.
Very interesting presentation. When you show like this composite model in which you basically print the you show that you can actually print two different materials, right? What is the resolution that you have, let's say, for example, in terms of the blood vessels that you can that you can get for this embedded, endothelial cells, like these vessels that you produce?
Okay, so that's a good question, actually. In terms of the resolution, if you look at it, the resolution is something that is mostly dependent on the material, because some material may just stay, once you lay them, they stay right there if they are really viscous. But something that is not staying right there, but it's kind of not viscous, but more, or let's say less viscous, then that is going to kind of reduce your resolution because it's going to kind of diffuse and fade. But the equipment itself has the resolution to go down to even 1 micron level, which you probably don't need. Now, by 1 micron, I don't mean that the nozzle size is 1 micron, because you really don't need a 1 micron, even though we can go down to 30 microns for materials. For whenever you use cells, you want to be somewhere in the range of 100 to 200 microns, because you don't put too much shear pressure on the cells. So the resolution of your printed structure can be, depending on whether it has the cell component or not, vary from tens of microns to close to 100 or 150 microns.
Okay.
Does that answer your question?
Yeah. Thank you.
So that is variable, and that simply depends on the material and your actual composition of cells or not cells in that particular spot.
Thank you.
But the equipment has the ability to go down to 30 plus microns, which is pretty decent, and which is actually in most cases good enough for modeling.
Thanks.
So as an extension of that, if you're thinking about, can we recreate capillaries? Say, for example, somebody is doing a kidney regeneration project, and they're trying to build a kidney by bioprinting. You can go down to some level of vasculature, but to go down to those ultra fine capillary, we're not there yet. Probably there will be other technologies. I'm sure a lot of people are thinking about those problems, because incorporating vasculature into printed tissue is a very hot topic in this space. They are thinking about other alternative approaches, totally out of the box ideas. So I'm sure those things will soon catch up and make this more widespreadly, more widely applicable for many other areas as well. We'll get that. But right now, you can just do the basic modeling using a basic type of vasculature and things like that. No problem. The real power and potential would be if you were to think about controlling the cell interaction and or the spatial distribution of cells within the tumor microenvironment. No other technique like your passive 3D cell culture on scaffold, none of those would would make that possible, because they are all self-assembly. You just drop the cells and then you assume they are staying there. You cannot control which cell goes. That's one advantage here, that you can control the distribution and complexity. Second, you can also do high throughput if you want. So those are all some advantages to take note of here with bioprinting.
Great. Well, thanks everyone for your time. Thanks everyone for joining, and hope everyone has a good rest of the day.